Hello. In today's video, we shall learn in detail about red algae Polysiphonia. By the end of this video, you will have a clear understanding about the structure of Polysiphonia, its modes of reproduction, its life cycle with triphasic alternation of generations, that is, two diploid phases, carposporophyte and tetrasporophyte, alternate with one haploid gametophytic phase. Let us first get an idea of the characteristics of class Rhodophyceae. Most of the members of class Rhodophyceae are marine. The multicellular forms may be filamentous, parenchymatous, feathery, or ribbon-like. Their mode of nutrition may be saprophytic, epiphytic, or parasitic. They exhibit a variety of colors due to the presence of various pigments in the chromatophores. Besides chlorophyll, carotene, and xanthophyll, water-soluble phycoerythrin is seen associated with blue pigment, the phycocyanin. The members of Rhodophyceae are eukaryotic and have membrane-bound organelles. The cell wall is made up of inner cellulose layer and outer pectin layer. Pits are present in cell wall through which cytoplasmic connections, plasmodesmata, are maintained. Assimilatory product, fluoridin starch, is also a characteristic feature of Rhodophyceae. Mode of reproduction may be vegetative, sexual, or asexual. Motile reproductive units are totally absent. Male reproductive organs are spermatangia, while female reproductive organs are carpogonia or pericarp. Sexual reproduction is always oogamous. Post-fertilization changes are highly elaborate. Members of Rhodophyceae show biphasic or triphasic alternation of generations. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us take up red algae Polysiphonia and study about it in detail. First, structure of Polysiphonia. It is a marine red alga belonging to the class Rhodophyceae. The plant is attached to the substratum with the help of long unseptate rhizoids. The thallus is dichotomously branched. The plant body is polysiphonous with many interconnected filaments. Red color of the algae is due to the presence of abundant phycoerythrin pigments along with chlorophyll A, carotene, xanthophyll, and phycocyanin. The thallus shows the prostrate system and the erect system. The prostrate system is creeping and is attached to the substratum with the help of rhizoids, which are single-celled and thick-walled. The erect system develops from the prostrate system and shows parallelly arranged siphons. The cells of each siphon are placed end to end and are connected by plasmodesmata. The siphons are of three types: central siphon, pericentral siphon, and cortical siphon. The cells of the central siphon are large and elongated and are centrally located. On either side of the central siphon are situated pericentral siphons which are narrow. They develop by the longitudinal division of the central siphon cells. These pericentral siphon in older branches may cut off smaller siphons periclinally on either side, which are called cortical siphons. The cells are connected by protoplasmic strands called plasmodesmata. At the apical portion of the thallus, only central siphon is present. The thallus of Polysiphonia shows apical growth, which is facilitated by a single dome-shaped apical cell. The thallus has two layered cell wall, outer pectin and inner cellulose. The cell is uninucleate with viscous protoplast, large central vacuole, discoid chromatophores without pyrenoid, and contains reserve food materials in the form of floridin starch. Next, we shall study reproduction in Polysiphonia. Mode of reproduction in Polysiphonia may be vegetative, sexual, or asexual. Vegetative reproduction is by fragmentation. When the plant breaks up into many pieces, each piece is capable of giving rise to a new individual. Sexual reproduction is of advanced oogamous type. The male reproductive organs are the antheridia and the female reproductive organs are the carpogonia. Development of antheridium. The male plant of Polysiphonia bears clusters of antheridia near the tip of the main filament. A central axis of elongated cortical cells is differentiated and is surrounded by pericentral cells in which the primary antheridial or spermatangial mother cell develops. Each one cuts off 2 to 4 unicellular spherical spermatangia. Each spermagonium develops into a single non-motile male gamete, the spermatium. Each male gamete is liberated through a narrow apical slit present on the wall and is dispersed by water. Development of carpogonium. Carpogonium arises from a female pericentral cell. The pericentral cell divides into a two-cell structure forming a lower basal cell or supporting cell and an upper carpogonial branch initial, which further divides to form a row of 3 cells. The uppermost cell of the carpogonial branch elongates to form the neck region trichogyne. The carpogonium is a flower-shaped body with a basal solan region where the female nucleus is located. Next are the pre-fertilization changes. Basal cell cuts off a cell towards its base known as the basal sterile cell. Sterile cells are also formed towards the side of the basal cell. The carpogonium is ready for fertilization at this stage. Adjacent to the supporting cell, the pericentral cells form sheath around the fruit body called the pericarp. Fertilization. The spermatia, which are dispersed by water, adhere to the tip of the receptive trichogyne. The wall between the two dissolves and the male nucleus makes its entry into the carpogonium through the trichogyne. The male and the female nucleus unite, forming a zygote. post-fertilization changes are highly elaborate in Polysiphonia. Several basal sterile cells and lateral sterile cells are formed. The supporting cell enlarges and divides transversely to form auxiliary cell between itself and the carpogonium. Protoplasmic connections develop between auxiliary cell and carpogonium. The diploid zygote nucleus divides vertically and forms two diploid nuclei. One of the nuclei migrate to auxiliary cell while the other remains in the carpogonium. The haploid nucleus that was present earlier in the auxiliary cell degenerates and only the diploid nucleus remains in it. The trichogyne is separated and later degenerates. The carpogonium, auxiliary cell and the supporting cells form a central mass called the fusion cell or the placental cell. A number of gonimoblast mother cell or gonimoblast initials arise from the placental cells. These gonimoblast mother cell form a number of gonimoblast cells. Each cell receives a diploid nucleus from the placental cell. The terminal end of the gonimoblast forms carpospores, which are enveloped within the wall of the fruit body, cystocarp or carposporophyte. Next, let us learn about the carposporophyte of Polysiphonia. In the life cycle of Polysiphonia, the diploid sporophyte phase is dependent upon the haploid gametophytic phase. The carposporophyte is made up of many gonimoblast filaments attached to the placental cell, which remains covered and protected by the sterile pericarp. The lowermost cell of the filament gives rise to the rhizoidal cell, while the uppermost cell forms the apical cell. The apical cell gives rise to the central siphon cell by undergoing transverse division. It also divides periclinally to form pericentral cell. The diploid carpospores are carried away by water and germinate on a suitable substratum to give rise to diploid tetrasporic plants. Now, let us study about the tetrasporophyte of Polysiphonia. It is a free-living diploid plant in the life cycle of Polysiphonia. The fertile pericentral cells first divide to form tetrasporangial initial and then divides vertically to give rise to inner and outer cell. The outer cell forms the covering by forming more cover cells, while the inner cell forms the sporangial mother cell. The sporangial mother cell divides transversely to form lower small stalk cell and an upper sporangial cell. The latter elongates to form tetrasporangium. The branches with the tetrasporangium become twisted and swollen. 4 haploid nuclei are formed by meiotic division of the diploid nucleus of the tetrasporangium. The haploid nuclei are surrounded by cytoplasm and develop into tetraspores, which are arranged tetrahedrally. The walls of the tetrasporangium ruptures and the spores are liberated. They germinate on a suitable substratum, giving rise to the gametophytic plants of Polysiphonia. Two of the spores germinate into haploid male gametophytic plant and two spores germinate into haploid female gametophytic plant. Finally, we shall summarize alternation of generations in Polysiphonia with the help of diagrams. Polysiphonia exhibits triphasic alternation of generations. They are gametophytic phase, carposporophytic phase, tetrasporophytic phase. Polysiphonia is a dioecious plant with separate male gametophytic plant and female gametophytic plant. The haploid male gametophytic plant bears sex organs, spermatangia, which give rise to haploid spermatia. The female gametophytic plant bears sex organs, carpogonium. Fertilization results in diploid zygote formation. The carposporophyte forms diploid carpospores in carposporangium, which give rise to diploid tetrasporic plant. The diploid tetrasporic nucleus of the tetrasporic plant undergoes meiosis or reduction division, giving rise to 4 haploid tetraspores. These tetraspores form the male and female gametophytic plant. Thus, two diploid phases, carposporophyte and tetrasporophyte, alternate with one haploid gametophytic phase. So today, we have learnt in detail about red algae Polysiphonia, showing distinct triphasic alternation of generations. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notifications of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.